Good morning, good afternoon, or good evening. Today we're going to talk about Chapter 10, and that is chromosomes, mitosis, and meiosis. During this video, we're just going to talk about chromosomes and mitosis. Meiosis is also in Chapter 10. I'm going to make a separate video for that one. Meiosis is involved in the production of gametes, sperm cells, and egg cells in mammals. This presentation is just about what are chromosomes and what is mitosis. Also, generally, we're going to talk about something called the cell cycle, which you can see right here. And that is essentially how we get one cell. It goes through its lifespan, and then at some point it gets some kind of signal to divide, and then it does divide. And that is that division. Part of that division is called mitosis, and it produces not one but two cells. And you can notice one of the cells kind of goes off here and does something, or this diagram doesn't discuss what it does. And then the other cell kind of continues to do the same thing according to this diagram. So this is the cell cycle. I'm going to start off with playing a video for you.
Cincinnati, Ohio is not exactly a place you'd expect to meet a medical miracle. But that's just what Lee Spivak is convinced he is.
And I put my finger and I said, you need to get rid of that airplane. And that's when I cut my finger off.
It just sliced it straight off.
Took it off. We don't know where the piece went. We never did find it.
How much of the finger?
It took off probably about that much of the finger.
The photos of his severed finger are pretty graphic. You can understand why doctors said he'd lost it for good. But it grew back with the help of a mystery powder. Nerves, tissue, blood vessels, skin, in just 4 weeks.
Miracle Man sprinkles miracle powder on his finger.
They've even called it pixie dust.
Exactly, they call it pixie dust. They sprinkle and it grew back.
Yeah.
But, I mean, was it at all like that?
That's exactly what I did.
This is where the pixie dust comes from, though at the University of Pittsburgh they call it extracellular matrix. They scrape the cells from pigs' bladders, dry out the tissue, and form either sheets or powder. Normally, when we're wounded, we scar, but the scientists say putting the extracellular matrix on the wound stimulates the cells to regrow instead. They hope the technique might one day work on organs and possibly more.
I think that within 10 years, I don't think that we'll be able to regrow a normal limb for you. I do think, though, that we will have strategies that will allow us to reconstruct the bones and to promote the growth of functional tissue around those bones, muscle tendon units. And that is a major step towards the eventual doing the entire limb. And that is a major step towards the eventual doing the entire limb.
Okay, so what you just saw in that video was essentially this guy's finger. It was cut off, so cut off, right? His finger is supposed to go there, and it was cut off. And normally what would happen is scar tissue would form. So scar tissue would form, but in this case, the cells began, the tissue regrew, including bone, including blood vessels, including muscle, including skin, the nail, everything. I believe even his fingerprints regrew to be identical to the original fingerprint. So what we're thinking, what we want to consider here is cell signaling. Cell signals. Normally, human beings are not capable of regenerating lost digits like this. Some other animals are. Some salamanders, if their tail is cut off, their tails can regrow. Also, some of their limbs can regrow. Even the entire limb can regrow. So what's interesting here is that normally, under normal circumstances, human beings will simply form a scar tissue, no new finger. But in this situation, they develop something from extracellular matrix, also called ECM for short, extracellular matrix. That's the stuff around cells. And it turns out that this stuff is really active in telling cells what to do. So something we want to consider is how do cells know what they are supposed to be doing? Are they supposed to be dividing? How long? How long should they continue dividing? What type of cell should they divide and become? This is a very promising field in biology. It's called stem cell research, and they have made some really incredible progress in this. If you are interested in looking at how third-degree burns are beginning to be treated, they've made a lot of progress with that, and that is basically that we're learning to intercept and manipulate cell signals and tell cells what they should do, what they should become, when they should divide, and when they should not divide. So there's a tremendous amount of medical implications for the ongoing research in this stuff. Okay, so moving on. To begin with, we're going to do a little bit of background, and it's really just a little bit. So I'm going to assign you a little extra reading. That's pages 68 through 69. We're going to come back to this, though, with more detail. The book order is a little bit odd. I would prefer to go over what DNA is and what RNA is first, but the book order is this, and I'm powerless to change it. So nucleic acids, that's pages 68 through 69. They're going to talk about DNA and they talk about RNA as well. Here, I'm only going to talk about DNA because it is the only thing that is relevant to mitosis. So please read section 3.5. It talks about RNA a little bit, but we're just going to talk about DNA in the lecture. So nucleic acids. Nucleic acids are what form DNA, and nucleic acid, let's get a better color here. This would be the nucleic acid, or a nitrogenous base, and we can see that there are 4 types. Over here, there's adenine, there's guanine, there's cytosine, and then there is thymine. So there's 4 of them. And we're going to come back to these terms. You don't need to worry about memorizing these things. Purines means there's 2 rings. So 1, 2, 1, 2. And then pyrimidines, that means that it is just a single ring structure. It's a chemical term, not that relevant to biology. So there's 2 types of nitrogenous bases, purines and pyrimidines, and they form what are called nucleotides. And again, this one we're also going to come back to a lot in greater detail in a later chapter, but you need to know a little bit about what DNA is for this chapter. So a nucleotide, that is what this is, nucleotide, right above here. Get rid of that. A nucleotide consists of 3 things. Number 1, a phosphate group. Notice it is negatively charged. There's a negative charge on the phosphate, just like the ATPs, there's negative charges there. There is a sugar, and it is ribose. Let's see, there's 1 carbon, 2, 3, 4, 5. So we could call it a pentose sugar. A little review from organic chemistry. So 1, 2, 3, 4, 5. It is a pentose sugar, and then this nitrogenous base. And again, the nitrogenous base comes in 4 kinds: adenine, guanine, cytosine, or thymine, and quite often biologists abbreviate that to A's, T's, G's, and C's. So if this was a new language, this language would consist of just 4 letters, A, T, G, and C, and this is what forms the code in DNA, the order of these bases. Now, a little more on that later. Here we can see more nucleic acids. Now the structure of DNA is as follows. Let's take a look at the top here. We can ignore this 5' for now. That'll be in a later chapter. We can see that there's a nucleotide here. There's a phosphate, and then connecting to our ribose or our pentose sugar here, and then a base. And we've now abbreviated it to a T. Now, there's something called complementary base pairing, and again, we'll come back to it later, but basically, T pairs with A, and these are hydrogen bonds. You can see hydrogen bonds. And then G pairs with C. G pairs with C. So T to A, G to C, pretty simple. And what this means is that we have, let me get a highlighter here, we have this nucleotide here on the left, and it is paired to a second nucleotide on the right over here. And so this nucleotide, again, same thing. It has a phosphate group, its sugar, and then its base. It's nitrogenous base here, which happens to be A, which is paired to T. And this forms what am I trying to say here? Two linear structures. One is here, and then the other is here. That's the word I was looking for. Double-stranded. There are two strands of DNA. One being here on the left, and then the other being here on the right. On the outside, we can refer to how we know proteins tend to behave in watery solutions, in aqueous solutions. These phosphates are negatively charged, so they orient outwards where there's more water. Negatively charged, they orient outwards. Then on the other side of the double strand of DNA, there's more phosphates here, and they're also negatively charged, so they orient outwards and can associate with water because they have a charge. The inside is these bases, purines and pyrimidines, well, just nitrogenous bases. That's all we need to know for now, nitrogenous bases, and they orient inwards. There's another diagram on the right here, and this one is, well, just the same thing without color. We have these sugars, and then we have these phosphates. And let's add one more term. This is called the sugar phosphate backbone. And why do we call it a backbone? Well, it is strong and it holds the double strand of DNA together. So phosphate to sugar, sugar to phosphate, phosphate to sugar, sugar to phosphate, and so forth. These are all covalent bonds. So covalent bonds are stronger than hydrogen bonds. The interior is held together, these base pairs, A pairs to T, G to C, T to A to T, C to G. These are held together by our old friend H bonds, and those are a little weaker, and they can be broken and reformed more readily. Next, when we have this double strand here, 1, 2, the final level of DNA structure on this slide is shown here. It is a helix, which is essentially a ladder that has been twisted. And then up here you can see it rotating in much, much greater detail. It is like a ladder, like a rope ladder that has been twisted. So it has a helical structure to it. Finally, leaving off, or beginning where we left off, with this DNA, which is a helix. We also would call it a double helix because there's two strands that are sort of rotating around each other in a helical manner. And DNA contains what we already know, certainly you've heard of this, genes, and that is the order of bases. So the gene number 1 could be A, C, C, A, T. There's no G's. But then there'd be a complementary strand here. So T to A to T, C to G, C to G, A to T, A to T. This order determines what type of gene there is there. So gene 1 is AACCAT on the left, and then gene 2 could be GACT, and then the other complementary strand, T to A, C to G, A to T, G to C. So the order of the bases determines the genes. Order equals genes. Okay. Over here, what we have is a picture of a chromosome, and a chromosome is essentially a really long piece of DNA. Very long. But why is it that it doesn't really look that long? It looks, in fact, kind of well, sort of fat and chubby. Let's wait till the next slide to actually get to why it is so chubby. Here it is. So chromosomes. Speaking of our chubby chromosomes, right here we can see sort of this X-like structure. I'll explain why it is in sort of an X-like structure, or kind of there's one thing. Let me get a more clear color. So there's one sort of piece of the chromosome here, then there's another sort of piece of the chromosome here. I'll explain what those are in a bit. But why is it this really thin molecule, DNA, over here looks really fat. The reason is that DNA is condensed. It is highly condensed. So here, this is DNA. Yay, DNA. And then here, we can see that there are sort of these ball-like structures that the DNA is wrapped around. And these are inside of there. Those are proteins. And proteins cooperate or interact with DNA. Specifically, they're called histone proteins. They interact with DNA to help it get packed tightly. And when we start packing DNA with proteins, we get a level of protein of DNA condensation called chromatin. Chromatin. This is actually from just a Wikipedia definition, just to cite my source there. I didn't put it in the hyperlink, but I want to read this one to you because I always find that I explain it differently every time. So chromatin is a complex, that means a combination of DNA and protein. It's found in eukaryotic cells, so bacteria don't have it, archaeans don't have it, prokaryotes don't have it. Its primary function is packaging long DNA molecules into more compact, denser structures, similar to somebody who is packing all of their clothing in their closet into a suitcase to go on vacation. You got to pack it to move it. This prevents these strands from becoming tangled, and it also plays an important role in reinforcing the DNA during cell division. And when a cell is dividing, the DNA has to be moved around kind of like a suitcase. So the DNA is packed really tightly, so when it's moved, this prevents damage during cell division. Also, we talked a little bit about this earlier, gene regulation, regulating gene expression. If you pack it up, so you pack your shirts into a suitcase, you can't use them when they're packed up. So when we pack up the DNA into chromatin, into really condensed chromatin, it can't be used. The genes in it cannot be expressed. So this is one way that we can make a cell become a specific cell type. We can pack some of the genes into chromatin. So perhaps if it is let's see, the guy was regrowing his finger, right? So he needs some more skin to grow the end of that finger's skin. So what would happen is all genes relating to kidney function would be packed or condensed, and that would be condensed, and that would regulate all the genes that are associated with kidney stuff. They wouldn't be used, so this cell would be a skin cell. So we can pack the DNA into chromatin, and that is part of how we turn genes on and off. Next, DNA replication. When we copy DNA, packing plays a role. Chromatin plays a role. During mitosis, that's the division of the nucleus, and meiosis, that's another type of nuclear division. Chromatin facilitates proper segregation of the chromosomes in anaphase. This part is quite impossible to understand, this last bit, until we actually know what anaphase is and segregation, so I won't talk about that one. So, the details of packing. We have right here a double strand of DNA, a double-stranded DNA. So we could see maybe if that's an A, then this would be a T there. If this is a G, then this would be a C, right? And then, okay, the blue one's a G, so, oops, and let's say G, there we go. G, okay, G, and then there'd be a C, right? So double-stranded DNA. Now we've simplified the double-stranded DNA just to a red line here. And what happens is DNA wraps twice around these proteins called histone molecules. And you need to know what histone molecules are. They are proteins. They are globular. You can see they're like kind of like a ball structure, and there is 8 of them. 1, 2, 3, 4, and then 4 more behind. So 8 total. And the DNA wraps about 2 times, it's like 2 point something times actually, around these 8 histone proteins, and that forms a nucleosome. a nucleosome, and that is the basic unit of packing for DNA. This last thing, the green fella here, is a linker. It's already written here, an H1 linker protein, and that just makes sure that the DNA stays wrapped around these 8 histone proteins when it is supposed to be wrapped around it. It can be unwrapped also. Okay. Next, I'm going to show you a video about all of this DNA packing so you can kind of visualize it. Here we go.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin.
Okay, the remainder of this video is beyond what you need to know for this chapter, but I'm going to play it until about halfway through, just so that, because it will help you to understand what we're going to talk about.
This fiber is then looped and coiled yet again. Leading finally.
Okay, so now what we are going to see is sort of that X-like structure. There's a distinct portion of the chromosome on the left and a distinct portion of the chromosome on the right. Just notice that.
To the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated.
So what you just saw there is on the left, one cell, and on the right, another cell, or actually just a nucleus on the left and a nucleus on the right. And the DNA, or the chromosomes, were segregated. That means divided in half. Half went to the left, half went to the right. And if you remember those 2 portions of the chromosome, kind of an X-like structure, one of them was divided and went to the left, and the other one went to the right. We'll come back to that in more detail later. Okay, so DNA replication is not something we're going to cover in this chapter, so I'm not going to show you that one yet. We'll come back to it. Okay, so the human genome, what do we have here? The human genome is 22 chromosomes. And notice that for each one, so here's one right here, there's sort of two. And one of them comes from the father, and the other one comes from the mother, right there. So we've got 1, 2. One comes from father, one comes from mother. Same thing for every single one of these chromosomes. Because of that, we are called diploid organisms. Diploid, di meaning two, ploidy meaning, well, like a set of chromosomes. So we have two sets of chromosomes. You get one set from mom and one set from dad. Next, we have one pair of sex chromosomes, and that is as follows. So you could inherit, you're always going to inherit an X from mom, and from dad, you could inherit either a Y chromosome, which would mean that you would be a male, or you could inherit an X chromosome from dad also, which would mean you'd be a female. So again, this would be from mom, always get an X from mom, and you can get an X or a Y from dad. This is a topic that we'll get into a little later on. You won't be quizzed on knowing which one comes from mom and dad in this chapter. But what we are looking at here is the entire set of genetic information in a single human being. This is what it looks like when we line up all of the chromosomes in a cell next to each other. Chromosome number varies by species, so let's take a look down here at the bottom. Human, we have about 3,000 megabase pairs. So megabase, that would mean that we'd have to add another 6 zeros on here. 1, 2, 3, 4, 5, 6. So let's see, that's quite a lot, about 3 billion base pairs. Remember, one base pair would be an AT in the middle or a GC. So this would be 2 base pairs right there. That's how many human beings have. And notice that some other critters have quite a bit more. A lily, for example, has 50,000 megabase pairs. An onion has 15,000 megabase pairs, so it's certainly not something that is exactly related to intelligence, at least, unless lilies and onions are hiding a secret intelligence from us. Not to mention chickens. Boy, they must be geniuses somehow. So no relationship between amount of DNA and intelligence or complexity even. What was there? Oh, no single cell organisms on here. Didn't include any. Chromosome number, 23 for human beings, 30 for a cow, 39 for a chicken. It really varies, and it doesn't really say much about the organism in terms of its complexity or anything. So here we go, the cell cycle. G1, this is called growth 1, and what happens here is the cell is going to increase in size. Why is it going to increase in size? Because division just occurred here. Division, cells divide into identical cells, great. So if you have just divided one cell into 2, then each one is going to be half the size, right? Half size. So G1 is growth and also maturing. That means becoming a specific cell type. So cells mature into a cell type. So it could be a skin cell, could be a sperm cell, could be an egg cell, could be a kidney cell. G1. In S phase is when DNA is copied. And so remember that we were seeing these chromosomes in sort of this two blob orientation, where there's one on the left and one on the right. The reason that is as follows. Before DNA is copied, it would simply look like this. So when DNA is copied, one of these chromosomes, that's one chromosome, it is copied. So now what we have is this thing, and these are called sister chroma. Oh dear, that's not very clear. Sister chromatids, and they are 100% identical. 100% identical. Right out. 100% identical because we have copied the DNA. DNA is copied, so each one of these chromosomes is going to be copied, and then we'll see these more X-like structures. Later on, I'll just abbreviate that to something like this. Look, two sister chromatids. This is one chromosome. After DNA replication, we have two of them, and they are 100% identical. Next, we go to G2, and G2 is all, G stands for growth in the first G phase, and in the second one, it is more like preparation to divide. And we'll talk a little bit more about this, but basically, for now, the cell prepares to divide itself in G2, and then we get all these words which are involved in cell division, and that is what we're going to talk about during this chapter. Something that we perhaps seem to be skipping here, when is it that cells actually do their job, such as, well, I've been talking about kidneys today, so when is it that those cells are going to do the work of the kidney, meaning they filter the blood? That is going to be in G1. This is when they perform their function. So at some point, G1 ends, and these cells are triggered by some type of cell signaling molecule, which you've talked about. They're triggered to enter S phase and copy their DNA, and this happens pretty rapidly. So something like 90%, 95% of a cell's life is spent in G1, where it is performing its differentiated function. Okay. So a little note on that last slide. This one is not included in your book. Remember that cells are not just dividing all of the time. That would be cancer if they're dividing all of the time. Most of the time, they divide and then they do something, like they act as a kidney, a heart cell, a brain cell, etc., except for one cell type, and those are called stem cells. And your book very annoyingly doesn't discuss them, but I'm going to discuss them because they're extremely interesting and important for you, especially if you're going to work in the medical field. You should at least know what they are, stem cells. That also means, unfortunately, they're not fair game for me to test you on, but they will help you understand this chapter anyway. What they are is this. They are undifferentiated cells. What that means is they don't have a function. They're just like a circle under the microscope, and they don't do anything. If you destroyed all of them, there would be no immediate effect because they're not doing anything. However, but obviously they do have a function at some point. When they are stimulated, they divide by mitosis to replace dead cells, to repair tissues, to provide new cells for growth, or sometimes new immune system cells, such as producing immunity after you get sick, those memory B cells, if you know anything about the immune system. That is what stem cells do. So the fellow who got his finger cut off, stem cells were triggered, and because they exposed the severed end of his finger to extracellular matrix, which apparently has some type of signaling molecule, his stem cells divided by mitosis to replace the dead cells, but not in the form of scar tissue, but in the form of replacing all of the lost tissues, and miraculously, the cells divided exactly as much as they needed to. They regrew the finger, but then they stopped growing. He didn't grow a 6-foot long finger. The cells somehow knew how to stop, which is pretty cool, I think. Okay, so let's take a look over on the right here. Look, it's a stem cell, and this is a terrible animation that I created. 30 minutes of my time, I'll never get back. Stem cell line. These are what we call pluripotent. It's another word I can't ask you to memorize because it's not in the book. Pluripotent means, though, that they can become some of the body's cells. So differentiated cells, that means they have a function. They have a function, like kidney cells, heart cells, etc. So when stimulated, oh, I forgot that I put that label on there because this is an animation. A zygote, for example, this then can become any and all type of cells. So this is a fertilized egg. A sperm has just fused with the egg cell, and this zygote can become anything. Here is a very dramatic simplification of the development of a human being. Totipotent can become all 200 cell types. So cell division occurs, and some of the cells, big simplification here, this is not at all the whole truth. Some of them become differentiated cells and have a function. The others remain as a stem cell line, and these stem cells serve to replace, as I said, look at the definition of stem cells, they serve to replace dead cells and repair damaged tissues. So we get some kind of stimulus here, and whoa, we get another cell division. So we need to get more cells that have a function, and then the other cell replenishes the stem cell line. So point being, mitosis. That's what's happening here. One of the cells goes and has a function. The other one, in this case, is going to remain a stem cell. And then this just keeps happening. And this is thought to be one of the main causes for aging, that these stem cells essentially get tired out. They may also suffer from mutations, especially if we're around toxic chemicals that cause changes in DNA, or if we are smokers, or if we drink alcohol. All of those are mutagens. Mitosis. Simple. That is the topic that we're now going to go over. Let me give you the definition for it. It is the division of the nucleus. So that is to say, it is not cellular division. And you will perhaps even catch me saying that it is cellular division and pause the video and say, Mr. McLeod, you're wrong. It's not cellular division. It's not. It is only the division of the nucleus. And it produces two nuclei, or so two nuclei, each of which has chromosomes that are identical to the parent nucleus. So we get one nucleus, it divides to produce two daughter nuclei, and each of them are identical. These three nuclei, well, this one divided, so it doesn't exist anymore, but these two are identical. I'm first going to give you a very brief overview of all of the phases of mitosis, well, pretty much all of them, and then we're going to go through them again in more detail. Okay, so let's start with the first phase, which is prophase. Pro means before. So prophase, number one, we can see that the DNA here has been copied. DNA already copied. And at the end of this, you should be able to tell that the DNA has been copied just by looking. How? Because there are sister chromatids. So it says chromosomes replicate. This should say replicated. They have already replicated. This happened in S phase already. So they have replicated here. And what that means is that first they'd all look like something like this. And then after they've replicated, they have more of this X-like structure there. So sister chromatids. Next, the centrioles, and those are basically a whole bunch of microtubules. They have moved to the poles, and the spindle fiber has been produced. You'll see its function in a moment. The nuclear membrane breaks down or degenerates. Where does it go? The nuclear membrane is absorbed. It's absorbed into the smooth ER. Smooth ER. It's absorbed into the smooth ER. The next phase is metaphase, and now you can see that the nucleus is totally gone, so there is no nucleus. No nucleus at this point. Metaphase, meta, like middle. All of the chromosomes line up at the equator of the cell. And yes, you want to know that term. That's the one you want to use to describe what happens in metaphase. They line up at the equator. Now, these spindle fibers that were beginning to get produced in prophase, they attach at the centromeres, and that's the center of these chromosomes, each with their sister chromatid. That's where the centromere is here. Centromere. I'll label one. See? Centromere. So these microtubules have attached to the centromeres of each chromosome. Next, during anaphase, the spindle fibers spindle fibers, that's these fellows down here, they begin to contract and they pull these sister chromatids towards opposite ends of the poles. So chromatids are pulled to poles by these spindle fibers. So poles of the cell, well, that's a pole, that's a pole. And let me erase that actually. That's not quite accurate. This here would be a centriole. A pole simply refers to the opposite side of the cell. So this region of the cell is a pole, and then this region over here is the other pole. I erased that because I didn't want you to think that this black thing here is the pole of the cell. That's not the pole of the cell. So anaphase. Last one is telophase, and at this point the nuclei reform. Now we have one nucleus, two nucleus, and plural is nuclei. The nuclei are reformed, and this phospholipid bilayer is formed and surrounds these chromosomes, originating from the smooth ER. Next, this cell, so this is just one cell here. That's one cell. Cytokinesis begins, and this is not part of mitosis. Not part of mitosis. And what happens is basically we get a whole bunch of proteins that encircle the middle of the cell, and then they contract like a rope like you slowly tightening your belt around your waist until pop, your legs walk off, and then your torso walks off. Oh, sorry if that sounded a little weird and creepy. Anyway, this ring contracts until you've got pop, two new cells. Great. So that is an overview of mitosis. Now some more of the details. Mitosis, G2 of interphase. So interphase, let me remind you, consists of G1, where the cell does its differentiated function. It acts like a neuron. It acts like a heart cell. It acts like a kidney cell. Then S phase, that's when DNA is copied. And then G2, this is final preparations for division. So this is what's going on right before we enter mitosis. Okay, so chromatin, that means that the DNA is fairly condensed. Notice that you can't see individual chromosomes. Can't see individual chromosomes. Where we are just at the chromatin level of DNA packing. What does happen? Centrioles, that means we have two of them. The centrioles divide at this point. So we originally just had one. They're oriented 90 degrees to each other. Each one consists of nine groups of 3 microtubules. Anyway, centrioles divide to form 2 centrioles because this one cell is going to become 2 cells. So everything in it, we need to make sure that the daughter cells have one of them because they can't just create it from nothing. Or at least many things they cannot create from nothing. The nuclear envelope is still present, and that's just about all of it. So a couple of other things. You do need to be able to identify what phase of the cell cycle a cell is in. This is in interphase. Why? Because you cannot see the DNA very clearly. That's because it isn't very packed. This is also still an interphase because you can see the nucleolus. You can see the nucleolus. You can also see the nucleolus here. If you can see the nucleolus, then that means that if you can see the nucleolus, we are still in interphase because the nucleolus is a region of more condensed DNA that is involved in gene regulation, and it's a whole tangled mess, and you can't divide the DNA, the chromosomes, if it's a big tangled mess in the nucleolus. So if you can see the nucleolus, then we're in interphase. Now we are going to go to prophase, and then there's also this prometaphase, which is something between prophase and metaphase. So during prophase, the nuclear membrane begins to break down. Nuclear membrane begins to break down. Let me write that. Nuc membrane breaks down. And also we can begin to see individual chromosomes. Can see chromosomes. You can begin to see individual chromosomes. And notice here, this is a real picture, you can see individual chromosomes, and also the nucleolus is not present. Nucleolus not present. And that's because all the DNA has been packed into its individual chromosomes, highly condensed, super coiled. All right, so that is about all of it. But no, let me just say one more thing. A kinetochore, we'll come back to that. That is just something on each chromosome that binds to these spindle fibers. You might be asking yourself, how is it that these microtubules grow from the centrioles? How do they grow from the centrioles and hit these kinetochores or centromeres or whatever? We'll come back to the details. How do they grow and hit them and not miss? The answer is that they do miss. They are blind and they're going in all directions. Whoopsie, I missed. And then when they miss, they simply depolymerize. If you remember microtubules, they consist of alpha, beta, tubulin dimers. And these dimers can polymerize, kind of like a bunch of people in a crowd that all of a sudden hold hands to form a big long chain. That would be polymerization. Equally, these people can stop holding hands, in which case we'd have depolymerization, breaks down back into its alpha beta tubulin dimers. So the centrioles keep shooting out these microtubules that we call the spindle spindle fibers in this context. They keep shooting them out until they have bound to all of the kinetochores on every pair of chromosomes, and also from each side. And that leads to metaphase. Oh, sorry, I forgot. I wanted to give you more details on how those microtubules attach to chromosomes. Okay, so what we have here is that thing right there, circle. I've drawn it on there because almost no diagram shows a kinetochore. That is a kinetochore. And then this region here is the centromere. Chromosome. Well, I got a lot of these things on there. So that's the chromosome. Then the other one's another chromosome, and they're two sister chromatids. 1, 2 sister chromatids. And kinetochore. Kinetochore is the protein that binds to DNA and the microtubule. Let me skip that. There we go. Finally, I always forget the order of these animations. The kinetochore is this fella right here. Each chromosome has 2 of them, one for each side of the cell. So in fact, there would be another kinetochore on this side. I'll draw that in. And then you'd have another microtubule that would come from the other pole of the cell and bind to this kinetochore. Eventually, when we get to the next phase of mitosis, these microtubules are going to pull on the kinetochores in opposite directions, causing the centromere, that's this yellow region here, causing the centromere, can't talk and write at the same time, centromere to divide. What the centromere is, let me add a few details to that. Centromere is a bunch of proteins called cohesins. And these cohesins hold the 2 sister chromatids together. Here's one sister chromatid. And then here's the other sister chromatid. And they're held together by cohesins. And if you remember those other diagrams of sister chromatids together, you notice that this thing right there, this sister chromatid and this sister chromatid, they are really snugly together, and that's because there are actually cohesins all along here that are holding them together to prevent breakage and tangling. It's just that there are a lot more cohesins right here in the centromere. More cohesins there. Okay. So next phase is metaphase. Metaphase comes after prophase. Prophase is when we got all these microtubules or spindle fibers successfully attaching to the kinetochores here. Once that has been accomplished, we've had all the kinetochores bind to a microtubule or a spindle fiber. After that, the sister chromatids or the chromosomes, you can use either term, are maneuvered to the middle of the cell or the equator of the cell. They're eventually maneuvered to the middle of the cell. You can see this one hasn't quite gotten there yet. It'll get there eventually. Under the microscope, this is maybe the easiest phase to identify. So this cell is in metaphase, definitely in metaphase. You can see all the chromosomes right here lined up the equator of the cell. Here, this is where we would have those centrioles that are spitting out these microtubules, forming the mitotic spindle. And this is the whole cell. No nucleus because the nuclear membrane broke down during prophase. This is in an animal cell. This is in a plant cell. How do I know immediately that this is a plant cell? Well, plant cells are generally more rectangular. Not always, but often. Animal cells are not often this rectangular. So here's metaphase. This location is metaphase, but this cell is in metaphase. Chromosomes are in the middle of the cell. Okay, or equator of the cell. The next phase of mitosis, we can actually see it right here, and you can maybe guess what is going to happen is each chromatid is going to be pulled away from the equator, and that's exactly what's going on here. The chromosomes are getting pulled in opposite directions towards opposite poles of the cell, and that's called anaphase. Anaphase, right here. The daughter chromosomes have divided. We could, if I zoomed in on one of these that's not the right word, during metaphase, they would have looked like this, and they would be connected by a centromere, and they would have a kinetochore on either side. Kinetochore, 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 kinetochore. So this kinetochore would be that, this kinetochore would be that, and the centromere has divided. Centromere has divided. Thus, each sister chromatid can be pulled in an opposite direction. This is anaphase. Over here, looking at a real picture of a cell, again, this is anaphase. Pretty clear that the chromosomes are getting pulled apart. It's also clear that they are not yet condensed into sort of like a nucleus-looking type of structure. This one is more zoomed in, and it's really obvious that this is anaphase. The last phase is telophase, and in telophase, the big one here is a cleavage furrow starts to form around the cell membrane. So this is the cell membrane, and a cleavage furrow made of proteins is going to slowly pinch the one cell into two cells. Also, the nuclear envelope begins to reform. It is produced by the smooth endoplasmic reticulum. Here, again, on top is an animal cell. This is an animal cell. Cleavage furrows beginning to form. You can see right here, cleavage furrow. You can see here, cleavage furrow. And a contractile ring will slowly pinch these this one cell into two cells. Here in a plant cell, it's a little bit different. You can see sort of a line in the middle, and that's not a contractile ring, because remember, plant cells have a cell wall that is very rigid and it cannot be contracted. So what they do is actually, they will just build, that's not how you spell build, there's an L in there, build new cell wall. So that's what a plant cell does. They just build a new cell wall in the middle until eventually you've got two cells. They build a new cell wall. The next phase that is not part of mitosis is cytokinesis, and that's where we get this cleavage furrow here that is composed of an actomyosin contractile ring, and you can just delete that actomyosin, okay? I'm not going to require you to know that level of detail, just this. Contractile ring. Contract, you all know what that means. Contracts, it gets smaller, tightens, and it will eventually pinch off into two new cells. This is how an animal cell divides, by cytokinesis. Down here, we can see a plant cell dividing, and what happens is vesicles will first gather at the cell's midpoint. Those vesicles will be transported there on microtubules. The small vesicles will then begin to fuse, forming larger and larger vesicles. You can see some larger vesicles forming there, right? And then eventually, these vesicles, they contain all of the necessary molecules and macromolecules to form a new plasma membrane and also a new cell wall. It forms from the vesicle contents. One last thing about cytokinesis is this. The definition of cytokinesis. This definition is the division of cytoplasm. And the result is two new cells. So division of cytoplasm meaning organelles. Each daughter cell is going to need some organelles, right? Like a rough ER, mitochondria. If it's a plant cell, they're going to need some chloroplasts. All those need to be divided. The cytoplasm itself, or cytoplasm, refers to the organelles and the liquid. Cytosol is just the liquid. So many words. So division of the cytoplasm organelles, and what was the other thing I was going to say? I think just was that it? And then two new cells. Great. All together now, interphase right here. This is interphase. Why do I know that? Because the nucleolus is still present, because there's still a nucleus, a nuclear membrane more specifically. I know that this is not G1 because the centrioles have divided. There's two of them. That is going to happen in G2. So this happens in G2, the end of interphase. Prophase, nuclear membrane starts to break down. We can see that. Fragments of the nuclear membrane. Sister chromatids have been produced as a result of DNA synthesis. That happened in S phase. Prometaphase, I kind of skipped that one earlier. Prometaphase is this one. I am not going to be, some books don't even mention prometaphase whatsoever, so I'm not going to be quizzing you on it, but what it is essentially an intermediate phase between prophase and metaphase. It's where the chromosomes, each with their sister chromatid, are negotiated into being at the equator of the cell, which is what happens in metaphase. So metaphase, they're all at the equator of the cell. This one's the laggard, kind of slow. Come on, get over there. Okay, there. Now it's there. Anaphase, the chromosomes are segregated, the centromeres divide, and the microtubules pull sister chromatids to opposite poles of the cell. Telophase. Telo means far in Latin or Greek, one of those languages. Telophase, the chromosomes are far away from each other. The nuclear membrane is going to begin to reform, produced by the smooth ER, and then a cleavage furrow begins to form as well. It'll eventually contract if it's an animal cell, and then pop, there's two new daughter cells. Anything else that I wanted to mention here? Nope, I don't think so. So this is an animation of mitosis.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Alrighty then. Just a note on something I kind of misphrased. I said the centrioles divide. You can say the centrioles divide. It is in fact more true to say the centrosomes divide. Centrosomes consist of two centrioles. That was from chapter 4. Consists of two centrioles. Okay. So, the cell cycle control. What is the importance of the cell cycle control? Well, again, so that cells know when to divide and when not to divide. So, number one, right here, G1, the S checkpoint, so that is right here. This is, remember, G1 is when the cell is either performing its differentiated function or when it is resting as a stem cell. So some type of environmental signal is going to cause this cell to say, it is time to divide because we need more cells. More specifically, it's the first key checkpoint, and it ensures that the cell has the necessary growth factors, nutrients, and enzymes to synthesize new DNA. Without the proper signals, the cell is ready to proceed. The checkpoint will not let DNA synthesis begin. So essentially it is, okay, is everything okay? Do we have all of the nutrients and enzymes? One of the things that would be critical here would be, for example, folic acid. That's present in high concentrations in a lot of things. Dark green vegetables is one. It's a coenzyme for the replication of DNA. It's one of those things that I would say probably almost every nutritionist is going to say pregnant women should eat a bit more of that because it's involved in the replication of DNA, and a growing fetus is replicating its DNA a lot, especially in the first trimester. So if everything is okay, then we enter S phase. And S phase is where we take each chromosome. So if this is one chromosome, we're going to replicate it, and now it'll be like so. And we have two sister chromatids. Two sister chromatids. This vocabulary is extremely important. Pretty easy for mitosis, but the next topic, meiosis, it gets a little more complicated. So two sister chromatids, that's what we would describe this as. We would describe this as one chromosome. That's one chromosome. Here's why it can become more tricky, though, is your book, and even I will occasionally call this one chromosome as well. The reason that sometimes we will do that is that it is, in fact, only one chromosome, because each of these is exactly the same as the other. It would be kind of like you having two copies of the same book. How many books do you have to read? Well, you just have one book to read. You just have two copies of it, right? They're 100% identical. So this term up here, two sister chromatids, is how we explain that DNA has been replicated and they are bound by a centromere in roughly the middle. Okay, the G2M checkpoint. This cell cycle checkpoint ensures that DNA replication is finished before the cell begins mitosis. If a cell has damaged or unreplicated DNA, the checkpoint will not let the cell undergo mitosis. Lesson here is many mistakes are made by cells. Many mistakes are made, and if enough mistakes are made, then we will produce a cell that is non-functional or malfunctioning perhaps, or in the worst case scenario, loses control of its cell cycle, which would lead to cancer. So if these checkpoints are malfunction enough, then it leads to cancer. Lastly, the third checkpoint, sometimes called the spindle checkpoint. This checkpoint occurs at the end of metaphase and prevents anaphase from occurring until all kinetochores are properly attached to spindle fibers along the cell's midplane. So what we want is for all the chromosomes to be like so at the equator of the cell, and the cell has various protein-based mechanisms for signaling, yes, all of the kinetochores have attached to a microtubule. We can now proceed to anaphase because we need equal division of the DNA. For example, if we had only a microtubule attaching to this side, that would mean both of these chromatids would go to one pole and be in this cell, and the cell that would form from this pole would be missing one chromosome, which could cause the cell to die, could also cause a whole bunch of other disease-related states. Cell cycle regulation. Your book talks about cyclin-dependent kinases. So firstly, cyclin, why do we call these things cyclins? Because they vary in concentration. So over the course of the cell cycle, you might have more cyclin or you might have less cyclin, then more, then less, as the cell cycle goes round and round and round. Dependent kinases. So kinases, remember what they do is they phosphorylate other proteins. So what you can think is this. Once we get enough cyclin, so here we've got a lot of cyclin, then these kinases are activated, and when they are activated, they are going to phosphorylate other proteins and cause cell division. So cyclin-dependent kinases, CDKs. That's over here. There we go. CDKs, cyclin-dependent kinases. Okay, where were we here? Firstly, number one, cyclin is synthesized and it accumulates. So starting right here, we'll call that start. It's an arbitrary start point. We could see the level of cyclin going up, and when we get enough cyclin, say right here, cyclin is going to bind to CDKs, cyclin-dependent kinases, leading to this, well, this is just the CDK, cyclin-dependent kinase, and now it's bound to cyclin. So it is now what we call active. And this is mCDK because it's associated with mitosis. And you your book mentions a whole bunch of other types of CDKs. Nope, you don't need to go ahead and memorize those. But basically, when it's activated, we have this mCDK. Cyclin has bound to CDK, cyclin-dependent kinase, and this can activate a whole bunch of proteins, written right here, phosphorylates the proteins, of course, that's how they're activated, and it activates the ones that are associated with mitosis, and it may also inhibit proteins that are stopping cell division. An example your book talks about is one of the proteins mCDK phosphorylates is P27, which inhibits cell division. There's a whole bunch of genes and proteins that inhibit cell division, and it's important that they are present because we don't want cell division to happen all the time, because that's called cancer, producing a tumor. So these CDKs will phosphorylate specific proteins at specific times when there's enough cyclin, and that leads to the loss of inhibition of cell division. The cell divides. Cyclin is degraded, but then it can also be reused and recycled for the next round of cell division. Cancer. Your book mentions the word cancer, I think, about 8 times and then never really describes what it is. Again, your book is really the end-all and be-all of what you will be quizzed on, but I think that a little discussion of cancer will firstly be relevant to you. Many of you are going into allied health careers in allied health, and so it's something you're going to encounter, at least many of you. That, and also about a third of us will at some point get cancer sometime in our lifetime, unfortunately. So what is cancer? It is the uncontrolled proliferation of cells to produce a malignant tumor. Malignant tumors spread through the body. If it is not a malignant tumor, then it will not spread, and we call it a benign tumor. But malignant tumors are associated with cancer, and if you get enough of them and they get big enough, they interfere with normal body functions. What causes cancer? Well, smoking, drinking, other unhealthy diets, getting too many sunburns, essentially anything that causes mutations, or, and root words here, mute means change, gen means to make, so make change in DNA. Carcinogens are simply mutagens that cause mutations in genes that regulate the cell cycle. So carcinogens, mutagens, they're essentially the same thing. They cause mutations. And down here, mutations in genes which control the cell cycle, so P27, there's other ones, P53, mutations in genes that regulate the concentration of cyclin, that regulate the concentration of CDKs. These mutations could lead to cancer. Common carcinogens, this should be an F, sorry about that, UV light, tar and tobacco smoke, X-rays, and again, mutations, what they do, I'm just trying to relate this to a little bit of what we've learned already. Mutation causes a change in the nucleotide sequence of a gene, so that means, like, is it AACG? If we get a mutation when we're copying our DNA, it could be a GCG, so there was the mutation, right? So if we get a mutation in the nucleotide sequence of a gene, which produces a protein, we could change it to a new allele. An allele is a different version of a gene, like blue eyes, brown eyes, and that's another one we're going to come back to in a later chapter. Biology is a very cyclical subject. We get an altered codon, that's something in DNA. We know about the amino acid sequence. If you get a mutation, it affects the primary structure of a protein, that is the amino acid sequence. That means we're going to have different primary structure of a protein, might have a different tertiary and quaternary structure as well. Mutagens, carcinogens, cause mutations which cause uncontrolled proliferation of cells. So again, it is normal to have cell division, and we should generally have cells. We should generally have a tissue that knows when it has had enough cell division. Certain genes called proto-oncogenes, this one you can just not remember that if you don't want to. These prevent cell division. They inhibit cell division. So they're going to cause cell division to stop, and you get the same number of cells. Normal cells. We had 1, 2, 3, 4, 5, 6, 7 cells there. 1, 2, 3, 4, 5, 6, 7 cells there. Right. We have the right number. This is what would have happened with that fellow's finger who got cut off. His finger regrew, these cells stopped dividing. Somehow they knew, or they were triggered by some environmental stimuli. However, if we get enough carcinogens, enough mutations, tobacco smoke, UV exposure, radiation, infections, viruses, HPV, for example, these can cause mutations which would take this proto-oncogene, and it now becomes something called an oncogene, which means that there is less inhibition of cell division. So we'll just say no inhibition of cell division. So the cells just continue to divide and divide and divide, and we get the formation of a tumor cell. Excuse me, there we go. Another kind of gene that regulates the cell cycle is a tumor suppressor gene, and these stop cancer by chemically sensing that the cell cycle is out of control, and they cause apoptosis. That means cell death. It's like an auto-destruct sequence. One way they do this is by producing a glycoprotein, and that would be right down here. This would be a glycoprotein. So here's the protein. We can see it as an integral membrane protein. It's also a transmembrane protein. And then here is a polysaccharide side chain. So that's that part, glycoprotein. Glycoproteins, or glycolipids as well, they're inserted into the plasma membrane. The immune system recognizes this as cancer and destroys it. So basically an immune cell, an immune cell would come along, bind, so it's got some kind of a receptor on it that goes, binds to it and says, "Oh, you have had too many mutations, your cell cycle is not regulated," and this cell, natural killer cell, for example, would destroy the cell. It would do that by insert a variety of things. I won't get into it. Tumor formation and metastasis. I'm going to go over this really quickly because it's very interesting, but it's not going to be on your exam. So real quick, tumors, rapid repeated cell cycles occur, each with a short interphase until many undifferentiated cells are produced by mitosis. And again, those malignant tumors metastasize, the benign tumors don't. So you'll just have one tumor, and then it'll spread via the blood to other parts of the body. If it's the brain, that's a really bad one. If it spreads to another place, makes another tumor, like in the heart, that'd be a really bad one. In the lungs or lymph nodes is a really common place. All kind of bad. And eventually they get big enough to interfere with normal bodily functions. If you download the PPT that I'll upload on Blackboard, you can right-click these links and then select Open Hyperlink if you're interested in learning a little bit more about metastases and cancers. This one is about cell signaling as well, which we've talked about. Almost there. Cancer. Which of these do you think would be cancer? This is a pap smear down here. So there's smears of squamous epithelial cells scraped from the cervix, pap smear, and then a doctor would look at it, and the woman, only women have cervixes, yeah, that's plural. doctor would look at it and determine, is this or is this not cancer? And I can tell you that the one on the left here is, drumroll please, this is not cancer, and this one is cancer. Why? These cells are mostly nucleus in there. There's not a lot of cytoplasm. They're packed tightly together, and they are also smaller. That's a little bit harder to tell. Oh no, you can tell that. The size of this cell versus the size of, say, this cell. They're much smaller, and so they don't have any function. They just grow and grow and grow and grow. Epithelial cells of the surface. This is called a teratocarcinoma, and terato in Latin means monster. This was removed from a testicle of a man, and think of what the testicles do. They produce cells, sperm cells, that are essentially totipotent. A sperm cell combined with an egg cell can become a whole human being, and cancer in this part of the body can lead to some pretty disturbing tumors. This is an arm. This is a hand and even fingers, and they will respond to stimuli. Over here, this is a tumor produced in an ovary, and it was removed. These are teeth. This is hair down here. Some of these things will even develop a pulsating like heartbeat and will even twitch or contract when poked. So kind of disturbing. And you can consider, why is it that these tumors produce such a diverse array of tissues? Well, consider the fact that what egg cells and sperm cells do is they produce all the tissues when they fuse, forming a zygote. So these cells, even when their cell cycle is unregulated, they still produce many different types of tissues. Okay, so what is mitosis anyway? What is the purpose of mitosis in the body? How are the parent and daughter cells alike? How are they different? And why is regulating the cell cycle important in a multicellular organism? You should be able to answer all those questions. If you can't, feel free to post on Blackboard and ask questions of the class or me. Last topic here, and I've removed it from the same order of the book, that is binary fission, and this is how bacteria, or actually all prokaryotes, divide. They divide by binary fission. So let's start at the beginning. We have a bacterial cell. It has its one looped DNA, looped chromosome. Firstly, we get 2 copies of the chromosome. The chromosome is replicated, so now we have 2 loops. Then we get elongation of the cell. So the cell just elongates, and then they produce a new cell wall and cell membrane in the middle of it. And then we get 2 identical daughter cells, 2 identical bacterial cells. That's called binary fission. No mitosis for bacteria. All right, that is all. And we'll come back with a second presentation on meiosis.